Now we'll shift to the capillaries. These capillaries are, just to remind, they are connecting tubules between the arteries and veins. More precisely, they are connecting tubules between the arterioles of arteries and venules of vein. So, two blood vessels present between the arteriole and venules, they are all called as a capillaries. They are a small, around 3 to 4 micron, but their diameter size may varies from 30 to 40 micron depending on the organ. That largest capillaries are found in a liver. Capillary word that everyone knows now, that capillary means hair like, very small. Now capillaries, they are single layer flattened endothelial cells. From the start of this blood vessel lecture, I'm repeating this sentence again and again, that internal lining of all the blood vessel is made up of endothelial cells. They are single cell thick. In between the cells there are some gaps, and these endothelial cells, through pinocytosis and exocytosis, make exchange of material across the internal lumen, that is across the blood to the surrounding tissue fluid. So capillary are the actual site where there is an exchange of nutrients and oxygen and nitrogen waste take place, and that take place through these endothelial cells. So in this diagram they have shown two endothelial cells entangle with each other to form a round ring structure with central lumen. That central lumen, this round ring-like structure with the central lumen. This central lumen is where through which the blood flows. And this is a one endothelial cell, and this is another endothelial cell. So these only the two cells, they form a complete ring of a capillary. This large violet color dot indicates the nucleus, and these invaginations in the cells indicate the pinocytosis or exocytosis process going on with the surrounding tissues and blood. Now, something around this endothelial cell is present, that is a basement membrane, which is not a, they don't have any nucleus. They are elastic fibers like structures. And from where they arrived from this fibroblast, that is pericyte. Pericyte is the fibroblast which produce these elastic fibers, which gives supports to these endothelial cells from the surrounding, and that way the capillary remain intact. Otherwise that could have been ruptured, and continuously these pericytes are synthesizing this basement membrane. And these pericyte you will see only in some kind of capillaries, that is called as a continuous capillary. When they are permanent and large size capillaries are there, they are only you will see these pericytes. Or other way, they develop, basement membrane develop and they erupt, and the endothelial cell also dies, and there is a synthesis of a new capillaries every time. Now in this electromicrograph of a continuous capillary, you can see what we understood from the previous diagrammatic figure of the continuous capillary. We can verify whether it is real. So here it is a large nucleus of a endothelial cell, and one arm of that cell, and this another arm of a cell, or that could be another endothelial cell portion. They form these junctions, and these are they plays very important role in a exchange, ion exchange and many nutrient exchange, oxygen exchange thing, that is called as a cell junctions, when the two cells fuse together here. they are not fused, they forms a very minute micro gaps, and that through which there is a many gates like system through which the nutrients and nitrogen waste exchange take place. Okay. Now, because of this folding of a endothelial cell, here it forms a lumen, hollow cavity, and this is the portion from where there is a blood flow take place. Now, where is that basement membrane? So see this whitish color portion surrounding. Okay. This whitish color portion is a basement membrane, and these are the that fibroblast periocyte cells which produce this surrounding basement membrane. Now here this photograph is very interesting. It is of a continuous capillary. This photograph is a section through the fat tissue. So all this whitish empty space, these are the adipocytes or fat tissues. And to nourish this fat tissue, there are capillaries, and the thickness of capillary is, you can see here, a single RBC. So the thickness of this capillary is only of a single RBC size. So this is capillary here. This is capillary, and this dark portion indicate the endothelial cells, and you can see these endothelial cells forming capillaries. They are supported with these perivascular connective tissues produced from the periocytes. So there is anywhere you will find there is a development of capillary, and basic ingredient of forming any capillary in any tissue at the smallest level is a formation of 2 or 3 entangled endothelial cells and supporting basement membrane. That is the basic components of a capillary, which forms a capillary in a tissue. And one important thing you can remember here, the capillaries are continuously developing. Throughout the life, we goes on developing that capillary in different tissues as our fat, suppose our fats increases. So that fat, as the amount of fat increases, that fat get innervated with the such capillary formations, and the fat is maintained and nourished and increased through this capillary. One disease you may have heard about the related with the capillaries, um, varicose vein. Yes, varicose vein, that is altogether related with the capillary disease related with the capillary, where that capillary get ruptured. Frequently, the capillary get ruptures, and the blood ooze out in the surrounding tissue fluid. Now, in this slide, these are the different types of capillaries in a body, that is fenestrated capillaries. Now, do you remember somewhere you learn about the kidney histology of kidney glomerulus? So this photograph is of a kidney section. Now this is a fenestrated capillaries color photograph of a section of a kidney through the glomerulus. If you remember histological structure of a glomerulus in kidney, somewhere in earlier classes you may have learned in a 12 that many blood vessels are surrounding, they are packed into the Bowman capsules. Through this capillary glomerulus, all that blood except RBC get ooze out into the Bowman capsules, which is then absorbed, resecreted, reabsorbed, and forms the urine. Now, these fenestrated capillaries are present in a glomerulus. They have fenestrated means they have large openings in a endothelial cells through which everything except RBC pass out. Here in this stained picture, that is stained with the hematoxylin eosin stain. They have shown a convoluted mass of that fenestrated capillaries in the glomerulus. Now we'll see next slide exactly what is that fenestrated capillary is. Fenestrated capillaries are already told that they have very large size pores in a endothelial cells, internal lining of a capillary. They have large size pores, and through which the major content of a blood and content of a tissue get exchanges at this level. So you will find such kind of capillaries present in a small intestine where there is an active exchange of a nutrient take place. Then endocrine glands where they um, pour hormone secretions into the blood, and the kidney where during the urine formation process they require to pour everything into the Bowman capsules. So where there is a large molecular exchange take place at that location in the capillary, you will find these fenestration types of capillary. These capillaries are more permeable than continuous capillary. In continuous capillary, they don't only have to exchange material at very small rate and with the particular ingredients, particular molecules and compounds are get passed from the wall of endothelial cells in a continuous capillary. Majorly the glucose kind of a thing and oxygen and that nitrogenous waste only take exchange of these three things take place in a continuous capillary, but in case of fenestrated capillary many more large size molecules exchange take place, so they have large pores directly to exchange the material through this capillary. So these are the one of a type of a capillary. Now these fenestrations of holes in a endothelial cells or capillaries you will not see in a regular light microscope photography. For that you need to see the scanning electron microscope photograph. So here in these fenestrated capillaries scanning electron micrograph you can see these endothelial lining, they have very small size pores through which that exchange take place, and the surrounding capillary is surrounded with the podocyte nucleus. So nucleus of podocyte and podocyte which form the basement membrane, and these are the endothelial cell nucleus and endothelial cell wall and cell wall, and through which that fenestrated exchange take place. Now next photograph we'll see that exact what is that gap is, and in that magnified view of a electron scanning electron micrograph of this capillary you will see exactly what is that gap is. Now this is a magnified view of earlier electron micrograph. Now here this red color arrow the basement membrane, endothelial cell, and these are the gaps. This F indicate the gaps. What about these red structures? These are the podocytes. These are the extensions of that podocytes. Podocytes which combine other tissue with the blood capillary. They are the podocytes, and they control the opening and closing of these gaps here. And this is the basement membrane. These are the endothelial cells. So here at this side there is a lumen of a capillary, and through this blood or nutrient get passed through the pore and through the basement membrane it enter into the gaps between the podocytes. So the fenestrations or pores are not only in a capillary but also they are there in a podocyte. Podocyte is a kind of cells which form these gaps uh, between the capillaries and the tissue. So this F arrow indicate the small fenestrations. These are the parts of endothelial cells. Here at this location there is a capillary. These are the podocytes and these are the invaginations of these podocytes, and again they form the gaps here. Okay, one more type of capillary is a discontinuous capillaries. Now the name indicate itself, and you can imagine what could be that discontinuous capillary means. The capillary as such it's in other way the capillary when they form a tube, but here there is no such clearly distinct tube is there, but tube of a capillary, but it is very discontinuous, and that kind of thing you will see in a liver. If you have learned somewhere the liver histology in 11 12, livers, you take a section of liver, you will find there are a lot of hepatocytes, liver basic tissues of a liver, but that tissues are in such a way arranged that they have large sinusoids or gaps between the hepatocytes. Between the layer of hepatocytes there is a large number of gaps are there, and their gaps are called as a sinusoids. And when blood flows through this sinusoid with very sparingly or very less number of endothelial cells lying, that kind of blood flowing passages are called as a discontinuous capillary. Again, in short, a small galleries form between the hepatocytes are when filled with the blood. Now they are termed as a capillary, but that is discontinuous kind of capillary. These sinusoids itself serve as a capillary. I hope you got what is that discontinuous capillary. Yes. And where you will find this kind of thing when in a liver, spleen, some lymph node, bone marrow, and some endocrine gland, you will find such discontinuous capillary, where the large amount of a tissue, same kind of a tissue is there, and that tissue is invaded with the sinusoids, gaps, and some number of endothelial cells, and they together they form a, they work like a capillary, so they called as a discontinuous capillaries. Now in this photograph you will get the idea of what is that discontinuous capillaries. Now see, all this dark color portion is of a tissue, that could be the hepatocytes or lymphocytes or any kind of a spleen tissue. Here we'll consider this is as a hepatocytes. So these are the mitochondria in a hepatocytes, and hepatocytes are doing their job of um, detoxification and metabolism of nutrients. And here is a gap, a sinus or hollow space. These hollow space are lined with the endothelium. So here you can see this is a lining of endothelial cells. Because of presence of endothelium in a sinusoid, we term it as a capillary. Now in between there is they form such gaps. That gaps are called as a space of Dice. It's a site where actual exchange of nutrients and exchange of material take place between the blood and the hepatocyte. I hope you are getting the point of discontinuous capillary. There is no as such a single layer endothelial cell. Tubules are formed, but that tubules is irregular, and all the hollow spaces present in a hepatocytes or liver spaces all are filled with the blood with some number of endothelial cells, and that is now termed as a discontinuous capillary.